and welcome back to another episode of Teach Me in 10. This series challenges scientists to break down their research into just 10 minutes, making even the most complex topics engaging and easy to understand. I'm Brie Foster, a science writer at Technology Networks, and I'm thrilled to introduce today's guest, Simina Petrovan, Principal Scientist in Upstream Process Development at AGC Biologics. With over 15 years of experience spanning early to late stage upstream process development for mammalian systems, Simina brings a wealth of expertise to today's discussion. In this session, she'll explore the critical roles and responsibilities of upstream process development, showcasing how process development supports clients in bringing novel and innovative molecules to market.
Hello, my name is Simina Petrovan, and I am a Principal Development Scientist at AGC Biologics in Seattle, Washington. The primary focus of my career has been upstream process development, from cell line development to stage 1 of process validation and post-commercial support of mammalian processes. Thank you for giving me the opportunity to teach you about upstream process development and how this fits into the overall picture of bringing a molecule to the market in this episode of Teach Me in 10. The topics I want to cover in my presentation today are as follows. Discuss what process development is and how upstream process development falls into this picture. And the second point is elaborate on the UPD workflows and expertise, and finish with a summary slide. High-level summary of the company I represent. AGC Biologics is a contract development and manufacturing organization, also known as a CDMO, with a global network presence comprised of 8 facilities and more than 2,500 employees. To date, the company served more than 175 customers worldwide. First, I want to start with the big picture. What is process development? Process development at AGC Biologics is made up of 3 functions. Upstream process development, also known as UPD, downstream process development, also known as DPD, and analytical and formulation development, also known as AFD. Each PD function has expertise in the development of the mammalian or microbial process. In illustration, UPD deals with the development and validation of the cell culture process. DPD, or downstream process development, covers the purification, filtration, development and validation of the process, and AFD is responsible for method and formulation development for the process. Each PD function has a set of deliverables to fulfill in order to facilitate the advancement of a robust process from bench to large scale. To narrow the focus of my presentation on upstream process development. As stated on this slide, the UPD role is for the seamless execution of a cell culture process from clone selection to transfer into manufacturing. The cell culture process includes seed train, production, and the harvest unit operations. What are some of the high-level deliverables for UPD? UPD offers bench-scale experiments to aid or support the IND and BLA submissions to regulatory agencies. UPD also provides post-approval support for commercial processes by executing bench-scale experiments using pre-qualified bench-scale models. The type of bench-scale experiments and support provided by upstream process development will be discussed in more details in the following slides. A quick reminder to the audience, this presentation is mainly focused on the process development of mammalian and microbial processes. So the mammalian cell culture lab, what kind of workflows do we have in the mammalian lab? Our work starts with cell line development. With the selection of the final clone, the process moves into the process optimization phase, where the operating space of the process is defined in typically bioreactors, which could be glass, disposable, or amber bioreactors. At this stage, the optimized process may also be evaluated in a pilot lab. More information on a pilot scale in the lab are to come. From optimization, the process progresses into stage 1 of validation. As part of this stage, the scale-dependent unit operations are qualified in bioreactors, and the results are compared against at-scale data. If there are any bioreactor scale offsets present, these are identified and they'll be incorporated into the next stage. The next step of validation is process characterization, where the process parameter ranges are expanded outside the normal operating ranges defined in the optimization phase. The proven acceptable ranges and the process parameter criticalities are defined at this point, and the process control strategy documentation is authored in support of the process performance qualification campaign, also known as the PPQ campaign or stage 2 of process validation. As noted on this slide, UPD provides additional support to the transferring team at AGC, the MSAT team, via documentation verification, impact assessments, and deviation support. Next, I want to highlight the workflows followed by UPD to support the development of microbial cell culture processes. As part of the fermentation development and optimization phases, we start with the microbial strain that is transferred into our facility. The microbial strain is tested for reproducibility at bench scale. If necessary, the process parameters are optimized and checked for fit into manufacturing. At this stage, the operating stage of the process is defined for preparation for large-scale manufacturing. Similar to the development progression of a mammalian process, the microbial process moves into stage 1 of process validation. During this stage, UPD supports with bench scale data via the qualification of scale-dependent unit operations and process characterization experimentation. During process characterization, the proven acceptable ranges of process parameters are set along with their criticality levels. The process control strategy document is also authored at this time to support the PPQ campaign. In addition, UPD supports the transfer of the process to the transferring team MSAT team with documentation verification, impact assessments, and deviation support. What is the purpose of pilot scale runs and how do they add value to process development? The pilot scale runs serve a number of functions. These runs are used to test the readiness of the developed process for large-scale manufacturing. The pilot scale runs may be used for toxicology studies per client needs, and the clarified cell culture material may also be used as team supply for downstream PD and analytical group for their development of the process. I would like to mention that the bioreactor sizes in the pilot scale lab are 100 liters and 500 liter subs. Slide. The presentation highlighted the role, deliverables, and expertise of UPD in the development of mammalian microbial processes at the Seattle site of AGC Biologics. This presentation also highlighted how upstream process development fits into the process development function of the company. The overall goal of PD is to develop and transfer robust biological processes to large scale to help patients live better lives. In conclusion, I would like to close my presentation and state that I strongly believe that process development contributes to the mission of the company to bring hope to life. I would like to say thank you for your time and wish you have a good day. Thank you.
Thank you for watching. This episode was sponsored by your friendly CDMO experts, AGC Biologics. Their mission is to bring hope to life by enabling life-changing therapies for patients worldwide, helping to create a healthier and happier future. With a wide range of services, AGC Biologics supports clients in bringing novel molecules to market with confidence. Thank you for joining us for another episode of Teach Me in 10. Don't forget to subscribe so you never miss out on more quick science insights. Until next time, stay curious, and we'll see you soon.